Hello everyone, and welcome to episode 65. This is the 5th episode in this wonderful series on the kingdom fungi, and today I'm going to explore the evolutionary history of fungus. It's a long history, or more than 1.5 billion years old, and an explosion in more complex fungi began around the same time as the Cambrian explosion, so these more complex fungi have a lineage that's about as old as all animals. Fungal history is also a mysterious history, because the vast majority of the time, a fungus's body doesn't persist long enough to mineralize. As a result, the fossil record is really sparse on fungus, which has thrown their early history and their ancestry into a fog of the unknown. Animal bones fossilize way easier than hyphea and mycelia and mushrooms, so the animal fossil record is much more extensive. Although this isn't to say that a fungus can never fossilize. They can, and they do, and we found quite a few of them and studied them, and we've learned a little bit about the fungal lineage, even though relative to plants and animals, the data from the fossil record for fungus is still painfully sparse. Fungal mycelia is soft. It doesn't have any hard parts. There's no shells, there's no beaks, there's no claws or fangs, there's no scales, there's no thick cell walls like you would find in a tree. Fungus are very soft, and they live in wet, warm, dark places, which is what you would expect of an animal that lives to break down dead stuff. These conditions are optimal for decomposition. So when the fungus dies, it's basically an organism that's composed of a loose assemblage of strings made of single cells, and with no hard parts, the fungus is going to decay incredibly quickly. Geological processes like petrification and fossilization take an extremely long time, sometimes millions of years, and only then under the right conditions. So then, what are the right conditions for a fungal specimen to become fossilized? I would imagine that the mycelia body would have to be quickly encapsulated somehow. I'm thinking like a typical mushroom, not like a microscopic fossil, which would be extremely hard to find in the first place. If you had a mushroom, it could potentially be fossilized if it was rapidly engulfed in sediment, or ash, or mud, or something that would surround it in a particle matrix. This matrix seals off the mushroom from oxygen and bacteria that would promote decomposition. The pressure would build up as more sediment was deposited, over and over again over millions of years, and the fungus could become a fossil through the standard petrification processes. The mushroom wouldn't decay, because it basically got flash frozen by the initial flood of sediment. Most fungal fossils occur when something like this happens to them when they're in a symbiotic relationship with a tree or a plant. Animals have also been fossilized, and fungal parasites within their bodies were fossilized right alongside them, or right inside them, I should say. The fungus gets fossilized in these cases because its host is getting fossilized. Both of them are being exposed to the same geological conditions. They're both being exposed to conditions that predispose them towards fossilization. One of the most ancient pieces of evidence for fungus is a small, branching microfossil in the shape and the size of a hyphea, and it's argued by its discoverers to be a specimen of the form genus Ornatophyllum. However, a lot of people aren't really convinced by this thing, and so they looked for more convincing evidence. Perhaps the most mind-blowing evidence of an early fungal ancestor comes to us as an incredibly ancient fossil from the Topania genus. A paper published in the journal Paleobiology examined this extinct genus. The abstract of the paper says that Topania morphology, quote, shows it to have been an actively growing, benthic, multicellular organism, capable of substantial differentiation. Most notably, its septate, branching, filamentous processes were capable of secondary fusion, a synapomorphy of the higher fungi. Combined with phylogenetic, taphonomic, and functional morphological evidence such as hyphal fusion, identifies Topania reliably, if not conclusively, as a fungus. Probably a sister group to the higher fungi, but more derived than the Zygomycetes." Unquote. Now, that was kind of a big mouthful, so let me break it down a little bit. The author is describing a deep-sea dwelling organism, a benthic organism, with a dynamic, branching, filamentous growth pattern, which is just like a fungus. It's just like a mycelia. Furthermore, they have traits of modern-day fungus, like septa between their cells, a filamentous growth form, and specialized branching structures coming off of the hyphaea. It has all of the hallmarks of being one of the earliest fungal ancestors, as the fossils are dated at over 1 billion years old. This is ancient, like extremely ancient, before the Cambrian explosion ancient. This would have been among the earliest multicellular growth forms to have ever evolved, only after the evolution of eukaryotic cells themselves, and then multicellularity. Although, to be fair, multicellularity actually evolved independently in a wide range of different lineages, fungus included. It was from these marine-dwelling, benthic, amoeboid organisms that the fungal lineage is believed to have emerged. One of the earliest ancestor groups of fungus are the chytridiomycots, or the chytrids, which are microscopic organisms with chitin in their cell walls, and who use an external, saprotrophic, absorptive method of acquiring their nutrients from biochemical detritus. These chytrids mostly reproduce asexually by releasing a cloud of zoospores. A zoospore is basically just a regular spore, but it's equipped with a little flagella, like a little tail, and this flagella gets whipped back and forth to help the spore propel itself through the water. A small minority of chytrid species reproduce sexually. They release gametes, which meet somewhere in space and fertilize. In some species, the gametes produced by each parent are the same size. These are called isogametes. Now, in other species, the gametes produced by the parents are different sizes, and this is called oogamy. In oogamy, typically the male gamete is small and mobile, and the female gamete is a lot larger, and it's relatively immobile. There's a couple other methods for sexual reproduction besides just gametes touching, which includes the physical conjoining of a mycelial limb from each parent, like a rhizoid or a bit of hyphaea. This conjoining of the mycelia creates a membrane-bound corridor for each parent to send their gametes, which meet somewhere in the middle of this tube, of this corridor, to fertilize. This then makes a growth that buds outwards, and it grows into a structure called a resting spore. The resting spore is like a bag or a balloon that's full of zoospores, and when it ruptures or when it opens on its own, the zoospores get released, where they can go find somewhere else to germinate. The chytrid mycelia is primitive, limited, and rudimentary, and in some species, they don't really have mycelia at all. They're just bags of spores. These chytrids are some of the earliest fungi, and they're known from samples that are more than 400 million years old. And yet, even though they're this old, we can already see a lot of these characteristic fungal traits, these cellular and morphological and metabolic traits that define the fungal lineage. These chytrid fossils are some among many species that have been found in rock samples that come from an invaluable source of paleontological and evolutionary data. In central Scotland, there exists a fossilized mass of sedimentary rock. This rock formed in the late Devonian period, close to 410 million years ago. It's an amazing rock formation because trapped within its fossilized mass are the preserved bodies of numerous fungus and lichens and plants, and even a few small animals. This rock is called the Rhynie chert, which is considered to be an example of Lagerstätte. A Lagerstätte is a sedimentary deposit that's rich with fossils that have been preserved in excellent condition. The Rhynie chert is a particularly famous example of Lagerstätte, as it's filled with organisms from the Devonian era. It's like a little ecological slice of time that's been preserved for 410 million years. It's like a biological time capsule. This timeline also suggests that these organisms were members of some of the first lineages to colonize dry land, as this rock formed just a bit more than 130 million years after the Cambrian explosion. The Rhynie chert itself formed when volcanic hot springs filled a local body of water with silica compounds. This caused the water level to rapidly rise, where it flooded the banks or the coast. The coastal ecology was really quickly engulfed in this silica-dense water. The matrix provided the silica that would slowly fossilize all the plants and animals and fungus that were caught up in the water. The fungus and the Rhynie chert are representative of some of the earliest originating lineages, like chytrids, glomeromycetes, and ascomycetes. The chytrids in particular can be seen parasitizing a type of primitive vascular plant called a rhiniophyte. Fungus from the lineages Zygomycetes and Basidiomycetes aren't readily abundant in the Rhynie chert, but for different reasons. The Zygomycetes probably existed, but they just weren't around in this particular geographic location, and the Basidiomycetes probably hadn't evolved yet. The largest organism fossilized in the Rhynie chert is called a Prototaxite, which is a member of a genus of terrestrial fungi that existed from around 430 to 360 million years ago, so they were some of the earlier land-dwelling fungus. On a molecular scale, the Prototaxite specimens resemble primitive fungi. The growth forms are composed of masses of two types of cell structures. There's relatively thick, tube-like structures, which weave and wrap around each other like strands in a rope, and there's relatively thin, fiber-like hyphae that exhibit heavy branching. These branching hyphae interweave with these tube structures to form the overall mycelial tissue, which was apparently sturdy enough to support a relatively massive body. These prototaxites were really large because they could grow mycelia bodies that resembled tree trunks. They could grow up to 1 meter wide, and they could reach 8 meters tall. Near the tip, they would make short, rudimentary branches, which would grow into lobes or clubs that presumably released spores. There's some really fascinating evidence that suggests that these ancient fungi endured parasitic insects burrowing into their flesh to make shelter. Little carved-out tunnels and insect-like patterns attest to this. And the presence of these tunnels in a wide range of fossils suggests that the prototaxites had endured these burrowing insects for millions of years. Now consider that, at the time, these fungal structures would have been the tallest things around. The tallest land plants were about 1 meter tall, and wood hadn't even really been evolved yet. So when tougher vascularized stem tissue was evolved, and when lignin emerged, the plants could then support the weight and carry the water to grow really tall as well. They outcompeted these prototaxite fungus, and the prototaxites faded into extinction. Meanwhile, the insects that had evolved the mouthparts for boring into the fungus into these prototaxites, they're believed to have shifted over to these woody plants. As they could burrow into the wood to make their shelters, the wood became a replacement for the dense fungal tissue. Recent data suggests that the prototaxites may not have been a pure fungus, but may have instead been some kind of early lichen. There's a whole number of lichens that have actually been preserved in the Rhynie chert, including a newly discovered genus called Winfernatia. This newly discovered lichen genus has internal nets of hyphae that are tangling up the photobiont, which is basically a cyanobacteria. This specimen from the Winfernatia is believed to be an early relative of the zygomycots, or the zygomycetes, which I'll get into in a few minutes. While lichens are well preserved in the Rhynie chert, there are a few other sites that have prospective lichen fossils that are even older, like the 551 to 635 million year old fossils in the phosphorite of Wang'an County in south-central China. All of these early lichen fossils tell us something important about fungus. They tell us that forming symbiotic relationships like those in lichen were a paramount part of the fungal lineage from the very beginning, or from close enough to the beginning to not really make much difference. I should clarify that lichens form when the species involved meet in an evolutionary confluence, where they can all coexist with each other in a mutually beneficial way. When this happens, it's called a lichenization event, and these lichenization events have happened multiple times all throughout the history of fungal evolution, and they'll presumably keep happening into the future. Also understand that evolution is dynamic, it's not permanent, and a lichen will only persist as a lichen for as long as it's able. It's possible for the species that compose the lichen to evolve out of balance with one another, or to have their symbioses disrupted on an evolutionary level, to the point where the involved species de-lichenize and evolve to grow and live on their own. The Rhynie chert is fascinating not just because of all of these individual fossils of fungi and lichen and plants, but because these fossils were all once living organisms, interacting in a dynamic, evolving, living ecosystem. Some of these ecological relationships are preserved in the fossils, and we can get some insight into how these organisms lived in their primordial world. For example, some plants have leaves that show bite marks or chewing damage and signs of healing and repair. Fossilized poop gives clues as to what diets of larger organisms looked like, even if we don't know what those larger organisms were. And the fossil selection gives us insight into the structure of the waterside community more than 400 million years ago. With respect to fungus specifically, the Rhynie chert shows fungal specimens clinging to the roots of plants in what appears to be an extremely early mycorrhizal relationship. Other fungal specimens seem to be frozen somewhere in a pathological rage, infecting and parasitizing species of plants and species of algae. Some of the fossilized poop was full of spores, and this suggests that fungal spores and perhaps plant spores were so common and so dense that larger organisms like animals could feed almost exclusively off of them, off of the spores. I briefly want to talk about the relationship between plants and fungus, because these two kingdoms have had a very contentious evolutionary history together. Plants and fungus are beautifully complementary. The plants grow tall, reaching high up into dry air and bright sunlight, where they can photosynthesize and produce sugars. Fungi, on the other hand, grow laterally. They grow on or within the soil and detritus, and they use their huge surface area to creep into impossible places and absorb as much water and mineral nutrients as is physically possible. On their own, plants and fungi are optimized for the production or the extraction of certain resources. The sugars that the plant can easily make with sunlight are harder to come by for the fungus. But the fungus can absorb water and nutrients way more efficiently than plants can. So together, plants and fungi can move a huge amount of biological resources by combining their optimized strategies. However, the exact nature of this combination of strategies depends on both the species of plant and the species of fungus. Some plants and fungi exist symbiotically, in a mycorrhizal relationship where the fungus is fed sugar in return for helping the plant get water and minerals. Many of these mycorrhizal relationships are extremely beneficial for all parties, and they have secondary benefits for other organisms in the ecosystem. For example, say you have some soil that's kind of thin, and a tree would not be able to grow in that soil. But if the tree is in a mycorrhizal symbiotic relationship with a fungus, that fungus might just give the tree the edge. It might just allow the tree to cling to life in this thin soil. Now, if trees can live here, if trees can move into this area, that provides a physical habitat to all manner of other organisms, like insects, arboreal mammals, and even weeds and parasitic plants. This beneficial mutual symbiosis has very deep evolutionary roots, going back in time to the earliest colonization of dry land. It's believed that species of fungus were the first organisms to creep onto the land out of the ocean, and through their growth and metabolic activity, they slowly broke down the mineral bedrock into granules and particles. They slowly created soils and sands. And then following them came the early plants out of the ocean onto a primitive and undeveloped landscape. It's believed that the early plants were only able to hang on to life and spread because of the benefits they got from their fungal symbiotes, from the fungi that had already colonized dry land and had already done their best to make it more accommodating for other life forms. However, this kind of paints a rosy picture, and plants and fungi don't have a perfectly nice mutualistic relationship. Where many species have evolved these kind of nice mycorrhizal symbioses, many other species have evolved some kind of parasitism. In very rare cases, the plant may parasitize the fungus, but in the vast majority of cases, the fungus will find a way to parasitize the plant, and the plant has to find a way to defend itself against the fungus. This dichotomy, this back and forth, creates a repeating pattern of the fungi evolving an attack, and then the plant evolving a defense, and then the fungi evolving another attack, and then the plant evolving another defense. And this goes back and forth for millions of years. This is the core mechanism of an evolutionary arms race, or a co-evolutionary relationship that shapes both groups. This dynamic provides a huge amount of selective pressure, fueling speciation and divergence in both the plants and the fungi. The parasitic fungi have evolved to engage in a kind of bio-warfare with their plant host, feeding off of them without necessarily killing them. Although I've talked about facultative saprophytes, which are species of fungus that will parasitize and then kill their host, and then they'll eat their dead body and use the nutrients to fuel the parasitization of other hosts. Now, in other cases of fungal parasitism, the strategy of the fungus seems to be not necessarily on killing the host, but instead on stopping the host plant from reproducing. Fungal infections have evolved to target the seeds and the flowers and other reproductive structures of their particular plant host. The strategy is that the fungus can scuttle the reproduction of the plant and interfere with the production of genetic variety in their host, which might include alleles or allele combinations that could infer traits for resistance against the fungal parasite. As the parasite, the fungus species is incentivized, it's evolutionarily pressured to find ways to stop or to slow the plants from evolving resistance to the fungal parasitism. So, to boil it down, their strategy is to literally try to stunt the evolution of the plants. Now, even among the standard brutality and suffering that's so common in the natural world, this is a pretty cold move. Alright, so let me shift gears here. Earlier in the episode, I talked kind of briefly about chytrids, which are some of the earliest true fungi, and which are known by their presence mainly in the Rhynie chert, dated to around 410 million years old. Obviously, the fungal lineage kept evolving and branching off, so what came next? Before I answer this, I want to clarify that most of the species I'm going to be talking about had already evolved by the time the Rhynie chert formed. By the late Devonian, by 400 million years ago, most extant fungal lineages had already emerged. They had already appeared and spread and adapted to their habitats and their ecological niche. With that said, first the chytridiomycots appeared, and then came the blastocladiomycots. The blastocladiomycots, or the blastoclads, make up an average to a relatively small-sized phylum, with some of the species sharing many similarities with chytrids, similarities like the ubiquity of asexual reproduction and a monocentric growth pattern. Other blastoclad species show a little more evolutionary advancement, with more extensive amoeba-like mycelium and the capacity for sexual reproduction by releasing or contributing gametes. Most profound, these blastoclads are the first fungi to exhibit a property of their life cycle called alternation of generations. I've talked about this phenomenon before in the plant series, and it also exists in an analogous format in many fungi. The basic gist of alternation of generations is that the life cycle produces this alternating diploid and haploid form, and each form produces the other form as an offspring. So a diploid form will give birth to a haploid offspring, and when it grows up, that haploid form will give birth to a diploid offspring, and back and forth. The lineage of plants, and of many fungus, walks back and forth between diploid and haploid, between sporophyte and gametophyte. I talked about this concept in the previous episode on fungal reproduction. To briefly recap that episode, the mycelia of most fungus is almost always haploid, and it chooses reproductive partners based on a chemical signature called a mating type. Two prospective parent fungi with compatible mating types will fuse their hyphaea, and their gametes are mobilized. In some species, the parents will create a whole new branching mycelia with dikaryotic cells, each cell in the new tissue having two nuclei, one from either parent. In other species of sexually reproducing fungus, in most species of them, I believe, one parent will donate their gametes into the body of the other parent, which then grows the dikaryotic mycelia as a branching part of itself, kind of like a literally pregnant fungus. In either case, this step is called plasmogamy, when the membranes meet and the cytoplasm mixes or fuses. In this dikaryotic mycelia tissue, reproductive structures called sporangia are generated en masse. They're generated on a huge scale. These sporangia have cells that undergo karyogamy. Where all the other cells in this new mycelia are currently dikaryotic with two separate nuclei, the cells in the sporangia undergo karyogamy to become diploid. These diploid cells in the sporangia undergo meiosis to generate haploid spores with some randomized genetic information. And these haploid spores are released, where they go into the environment and they land somewhere and germinate and become new mycelia. You can see the alternation of generations here, even though it's very, very short. In these species of fungus, the diploid stage is a short-lived mass, no larger than a few cells, which has the singular purpose of producing more haploid spores. For comparison, think of plants, where all of the huge trees and stuff that you see outside are pretty much all diploid. Okay, so to tie all of this back into the original point, this mechanism of alternation of generations emerged in a blasticlad ancestor, which shared this quality with all of the rest of the fungi. The next branches that emerged in the fungal lineage included a small group of plant parasites called the Olpidiales, no doubt encouraged by the spread of plants onto dry land. If plants spread out and diversify, that's all the more plants that you can diversify to parasitize. After them emerged the entomorph Thoromycota, and after them the Kixellomycota, which both had species focused not so much on parasitizing plants, but on spreading throughout this new soil. Traits for saprophytic feeding were encouraged and spread throughout these new populations, and many of these species in the soil began to parasitize the insects, the rotifers, the nematodes that they found crawling around with them in the dirt and the mud. Now following these groups came the Mucoromycotina division, which contains 3 orders of more saprotrophic and parasitic fungi. This includes many species of the well-known zygomycots, which use trisporic acid as a pheromone to attract mates of the opposite mating type for sexual reproduction. These zygomycots are named after the zygosporangia that they produce, which are reproductive structures that create globular or spherical spores at the end of strands of hyphaea. When you see a zygomycot mold growing on a piece of fruit, for example, and it's undergoing sexual reproduction, its body will probably look like a bunch of thin little hairs, all lifting upwards off the fruit, with little dark balls or spheres held up at the tips. These spheres produce one or more spores, which get released to form new mycelia. The next emergent lineage was the glomeromycots, and this emergence was hugely important to life on land. The glomeromycot fungi are entirely asexual, and they're almost entirely unable to live on their own. Many of these species require a symbiotic relationship with a plant in order to survive. This symbiotic relationship manifests itself in the form of arbuscular mycorrhizas. In this symbiotic growth form, the hyphaea of the glomeromycot fungus penetrates the root cortex, reaching between its cells to spread through the tissue. The hyphaea will penetrate the cell walls, but they won't penetrate the cell membranes. Instead, they'll push against the cell membrane from the outside. They'll push into it, creating a pocket or an invagination. If you were inside of the plant cell, you would see the membrane flex inward as it was stretched, but it wouldn't break or get punctured. This creates a hyphaea that has its tip pushed into a cell, wrapped in membrane. And this membrane-bound tip of the hyphaea can thus engage very easily in nutrient exchange with the cell. This mechanism sets up the basis of the arbuscular mycorrhiza relationship, which has enabled the host plants to colonize landscapes that they would otherwise struggle in. The last two groups, which are the most recently emerged in the fungal lineage, are the basidiomycots and the ascomycots. These two groups both have the same general reproductive strategy, but with slightly different structures involved. The ascomycots evolved structures called an ascus, or an asci for plural, which is like a long, thin tube filled with ascospores. The ascomycots are also called sac fungi, so named because the ascus is like a sac, or a bag for the spores. Now the basidiomycots evolved structures called a basidium, or a basidia for plural, which is like a thick club that holds the basidiospores. The basidiomycots, and to a lesser extent the ascomycots, all possess species that develop mushrooms or other large fruiting bodies. The oldest evidence of these mushroom fruiting bodies is around 100 million years old. For a long time, the oldest known mushroom was a 90 million year old fossil. Another fossil, about 99 million years old, was discovered later. And the most recent find is also the oldest. It's a fossil that's around 115 million years old. These estimates suggest that the mushrooms as we know them were first evolved sometime when dinosaurs still roamed the Earth. It's interesting to note that as the biological world diversified, more niches opened up. There were more species out there doing stuff, generating this and that biological byproduct, and performing particular ecological functions. To the fungi, who were the first organisms to colonize new landscapes on the early Earth, there were now much more species for them to prey upon, more species to feed off of, to parasitize, more species to form symbioses with, and all of this evolutionary opportunity led to a huge diversification of fungi over the next hundreds of millions of years. There were other events in the history of earthly evolution that also helped propel the diversity and the abundance of the fungi. The events that I'm thinking of specifically are mass extinction events. Mass extinction events, whether global or localized, tended to be followed in the geological record by a brief burst of fungal growth. Fungal spores are anomalously overrepresented in the fossil record immediately following mini mass extinction events. This isn't absolute, it's not global, but there's enough evidence here for us to kind of identify a trend. And if you think about it, this is something that we might expect. Think of a mass extinction event. There tends to be some kind of external abiotic variable that's affecting life on a mass scale, like shifts in temperature or oxygen content in the atmosphere, or a meteor impact that vaporizes a chunk of the planet's crust. These environmental problems will tend to kill off a lot of sea life and a lot of plants, and this will then lead to the death of terrestrial herbivores and carnivores. These events are marked by widespread death at every level. Populations will fracture and species will go extinct. Communities will dissolve and collapse, and ecological relationships will break apart. Everyone's fitness will drop to near zero in a world that seems to be thrown into chaos by this extinction event, by whatever triggered it. Now, if you're a fungus, and you can survive whatever these environmental changes are, this mass extinction event is potentially going to be pretty awesome for you. I mean, think about it. There's going to be endless detritus for you to consume, because everything around you is dead. The predators that eat you are all dead. The competitors that you have to fight with to get nutrients are probably dead or extremely reduced in number. In these conditions, fungi can grow to a massive extent, operating as the planet's biological recyclers, taking the uncountable mass of recently dead organic matter and dissolving it back into base nutrients. It's almost as if these extinction events are like massive battles between living organisms and the non-living environment, and the living organisms pretty much always lose. After every extinction event, the fungus will roam around the battlefield, walking among the corpses, and slowly cleans them up. The fungus will dissolve the detritus, they'll fertilize the soil with nutrients, and they'll make the bed, so to speak, for the eventual recovery and radiation of life. That is about all that I wanted to cover today for fungal evolution. Like I said at the beginning of the podcast, fungal evolutionary history it's a long story, because the lineage is absolutely ancient, and it's shrouded in mystery because of the relative lack of fossils. And it's complex, because fungi are complex. They're hyperdynamic, multifaceted organisms with 1,000 different approaches to life, to nutrition, and to reproduction. If you're enjoying this series on fungus, then be sure to check out the next episode, where I'll be exploring the deeply rooted relationship between humans and the fungus that share this planet with us. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.